Good evening. Welcome to our first Hudson Alpha Unite of 2024. And before we get started, humor me, please. I do that partly because I love looking back, and this view right here never gets old. And it is such a wonderful reminder of how great of a job I think I have that I get to be here and talk to you guys tonight, but also just how special of a place Huntsville, Alabama is. And how special of a place Hudson Alpha is, that we have the opportunity to come together on a Tuesday night and talk about science. And that I get to stand up here and tell you guys stories about science, which is just the most fun job in the world. So, y'all ready to jump into some science tonight? Everybody's got their cookies, you're fueled up, brain fuel, good. There are a ton of familiar faces in the audience, but for anyone who doesn't know that we haven't had a chance to meet, my name is Kelly East, and I get the immense pleasure of leading the educational outreach team here at the Hudson Alpha Institute for Biotechnology. And the Hudson Alpha Institute for Biotechnology, well, hopefully you know what that is, or at least you were interested enough to show up on this Tuesday night. But here at Hudson Alpha, we're a nonprofit genomics research institute that is founded in Huntsville, Alabama, with the mission of using the power of genomics to improve life. That's a really big, lofty goal, and you're going to hear some stories tonight of work being done at Hudson Alpha that shows that we are actively working towards fulfilling that goal of studying DNA and uncovering clues in the book of life of humans, of plants, of bacteria, and using that new knowledge to ultimately make life better for us, our health, for the earth that we're living on. And the way that Hudson Alpha does that is this really fantastic combination of teams that work at Hudson Alpha that are trying to move research forward, that are scientists doing the research, companies that are taking those discoveries and moving them to market, because that's the glue that kind of connects the discoveries that are being made to actually improving life, is taking that knowledge and doing something with it. And then the education team that I get the pleasure of leading, that our job, why we show up to work every day, is to connect with you guys, the community, to share science, and to inspire that next generation of students that are going to be the STEM professionals of tomorrow. At Hudson Alpha, we focus on studying the DNA largely in two domains. Humans, so all of us sitting in the room tonight, we all have DNA inside of us that we can research and help make discoveries that are going to improve health and disease, diagnostics and treatments, as well as scientists at Hudson Alpha that are trying to uncover clues in the genomes, in the DNA of our plants and our agriculture fields. And we're going to talk through some stories tonight in both of those areas, because what do they have in common? DNA, right? So the way we're going to do this tonight, and I say that one of my favorite things about my job is getting to talk to people about science, about the amazing discoveries that are happening here at Hudson Alpha and all across the planet. If I think backwards to when I was, maybe 13, 14 in school, I had some amazing teachers that got me hooked on science at a young age and told me stories about the discoveries that were being made, and that immediately wanted me to go into science. And I thought for a minute that I wanted to be making those discoveries and working in a lab. Those teachers did a good job of inspiring and training me to work in science. But what did I do? I went off to college, I jumped into a laboratory, and I learned pretty quick that I like talking to people more than using pipettes, which is okay. I'm so glad that there's tons of people that really do enjoy working in a lab. That was not meant to be me. But what I'm passionate about is learning about science and thinking about how is that going to impact the world around us, how is that going to impact us as human beings, and then getting to have a job where I get to help other people understand those things too. And that is just the most fun. So every year in HudsonAlpha Education, we publish this book called the Annual Guidebook. There's a whole bunch of pictures. It kind of looks like a pretty rainbow up here. These are all the past issues. We also have a few physical copies that we print every year. Make sure we get these in the hands of all the science teachers in our state so that they can take this back to their students and hopefully inspire more students to go into STEM and get excited about what's happening. But every year, we on our education team, we scour the literature. We read paper after paper after paper, and we pick out our favorite stories and we write them up for people to read in a way that's easier to understand. It's also a whole lot shorter than all those papers that we read, but we compile them into this guidebook. And so there is a brand new guidebook that literally got uploaded in digital form about 5 hours ago. How's that for hot off the press? So the QR code that's on the handout as you came in, that'll take you to that digital version. But the stories that I want to tell you tonight are all stories from that guidebook. So you can go read more about them if you find them interesting, and also know that there's a whole bunch of stories in there that we're not going to talk through tonight as well. So let's jump into some of these stories, and I'm going to frame this around what are the things that we've learned in the last year that we didn't know before, that these are stories that I couldn't have told you in this same talk a year ago. And we'll think and dream together about how far we've come in a year, and also think about what we might be talking about next year that the scientists are actively trying to figure out in the labs today. So based on the scientific discovery and the research that happened in the last year, we now have a more complete picture of what it looks like to be a human in terms of genetics. We have a more complete picture of the human genome. Many of you might remember, like I do, reading and seeing some of these images crossing your newspapers and your magazines back in 2003. These are both pictures that show that hallmark event of the publishing of the human genome for the first time. This big international consortium worked together for 13 years to sequence a person for the first time. 13 years. We now can sequence a genome in about a day. We've come a long way. But this was a huge, momentous accomplishment for science of publishing a human for the first time. And ever since that got published, scientists have continued to make it better. They've continued to study more and more people, help fill in the gaps that existed when they published it for the first time. It wasn't perfect, and have tried to iterate on that. And the reason that we need this is for scientists, as they're studying whatever question they're trying to answer, a lot of times they need to know, what does a typical human sequence look like? If they're trying to figure out what are the genetic changes that cause disease X, well, they need to be able to identify the differences. So they need to know what typical is. And so we use this human genome reference sequence as that kind of typical copy. Now, one of the limitations that exists with our typical human genome reference is that largely that data came from one person. Something like 70% of it is from one specific person. All of it is from a relatively small number of people, and they're individuals who answered a call in the Buffalo, New York newspaper in 1997. So, Buffalo, New York, 1997. How well do you think that represents the diversity of humanity on the planet Earth? Not great, right? So huge, huge accomplishment. But now that we have the ability to sequence more people faster and cheaper, we have the ability to do better, to create databases that scientists can use that better represent the diversity on the planet. And so that's what happened this past year. We now have not a human genome, we have a human pangenome. The human pangenome is a reference, it's a database that scientists can use that is, again, trying to catalog what typical genetic variation exists between people. But instead of being mostly from one person, this that they published this year is from 47 individuals from every continent on the planet Earth except Antarctica. There aren't people living down there to go get samples from. But it much more broadly represents diversity. And so they've published this as a resource for scientists to use. And by doing that, they have found that humans, there's a lot more diversity that exists than we thought existed. That of the 20,000 genes, recipes that we have inside our DNA as humans, more than 1,000 of them have these kind of sizable duplications, extra genetic information in them in some people. That is probably just normal variation, but now we know about that. And so the map of the human genome looks more like this than a straight line, where all throughout our DNA, there are areas where people can have extra letters or be missing letters that might have absolutely no consequence at all, or maybe it does. But the only way we're going to figure that out is by sequencing a lot of people and figuring out what is normal variation and what are the genetic changes that actually have an impact on a person. Now, they're not stopping at 47 people. This is again a worldwide consortium doing this work. They've got goals to take that number from 47 up to 350 people by the middle of this year. So rapidly evolving and getting better and better, and is a incredibly valuable tool in the toolbox for scientists that are doing human research, including our scientists here at HudsonAlpha. So those are people, and I also want to point out, as we're kind of walking through these slides, anytime that you see this HudsonAlpha logo over here, that points to the fact that some, at least, of the research that we're talking about was done by scientists at HudsonAlpha. I mentioned that HudsonAlpha is really interested in thinking about the DNA of people, but also plants. And this story is kind of similar to the last in that it is the creation of a better, bigger data set for scientists to use. But in this case, we're talking about plants. And so here in 2024, we now have a more complete picture of plant genomes, genomes being the entire genetic code of a particular organism, and a better idea about what the function is of plant genomes. What is the DNA doing inside those plant cells to control the plants, how they look, and what they do? So the work I want to tell you about is some work that was creating something that is called the Plant Genome Atlas. So again, this is a tool. Researchers can go and download this data and use it. And it is data from 17, of which most are pictured up here on the screen, 17 flagship plants that are really important for plant research. And so they picked 17 plants that are really important, and they created this atlas. This is work that was done by this group called the Joint Genome Institute. Collaboration is going to be a key theme tonight, that none of this work is being done by a single person, and a lot of this work is being done by scientists that are not even in close proximity of each other. The Joint Genome Institute, funded by the Department of Energy. You can imagine they've got heavy stakes in helping move science forward and thinking about what that can do for our energy needs. But this atlas is bringing together information about the DNA, the genetic recipe of all of these plants, but also the RNA of these plants. All right, so we're going to pull back to biology class for just a minute and think about how DNA and RNA work together. So we talk about an entire organism's DNA being the genome, the DNA or the collection of RNA from a particular cell is called the transcriptome, which is a giant word. But what happens, how our cells use our DNA is by transcribing, that's that transcriptome word, transcribing those DNA recipes into RNA. So by looking at the RNA at a particular point in time, you can make some, you can understand what parts of the DNA are being used, what's being turned on, what's not being turned on, which can be really interesting if you're trying to figure out what on earth this gene does. If you've got a gene you don't really understand, you could take a plant and you could do something about its environment, increase nitrogen, increase drought. And you look at how is that plant using its DNA differently in those different scenarios, which genes are being turned on in this situation versus this situation, and that can help you figure out what those genes are doing, what their job is based on how they react to certain environments. And so this database, this atlas, is pulling that data together for these 17 flagship plants and then updating it and pulling all that data together so that if I'm a plant researcher, I don't have to go scour the literature and pull the data from all of these different studies. I've got a one-stop shop where I can go and get this data that is consistent that I can use in my research. So these last two stories are stories that are going to help accelerate science going forward from here and let us move forward a whole lot faster because of these better tools and databases. The next story I want to tell you is about, so that last example we're talking about, sometimes you have genes you don't understand, you don't know what they do. And in science, we're trying to figure out what their function is, what that gene's job is. And some research from HudsonAlpha this past year, we were a part of a team that helped discover that a particular gene in humans is involved in brain development. So we think, we sequenced the human back in 2003, right? We thought we had it all figured out. We didn't. We still are really good at reading the DNA, but there are still big gaps in our understanding of what the DNA does. There are still lots of genes that are not well understood. And so what that leads to is a whole lot of patients that come and have genetic testing and end up with uncertain results or negative results because we aren't able to understand their DNA well enough yet. And so I want to tell you the story of kind of how this discovery happened. This is the paper. I told you that when we put together these guidebooks, we do a lot of the heavy lifting of reading articles with titles that read deleterious protein-altering variants in the transcriptional co-regulator ZMYM3 in 27 individuals with a neurodevelopmental delay phenotype. That's not the title in here. The title in there is New Neurodevelopmental Gene Discovered, right? But this was work from a whole bunch of people all across the planet as well. The names that I just highlighted up on the screen, those are HudsonAlpha employees. So these are our scientists that help move the science forward and help make this discovery. But this particular screenshot I pulled, this is only a few of the authors. It actually says author list continued on the next page. Collaboration is how discovery happens. But here's the story about how this discovery was made. So a patient shows up in a clinic looking for a diagnosis. This is a patient who has a neurodevelopmental phenotype. And when we say neurodevelopmental, we're talking about differences in how our brain developed. That can look like things like an autism diagnosis or developmental delay or intellectual disability. Those types of symptoms are often what you're thinking about when you hear neurodevelopmental. So this patient has developmental delay and also has some specific differences in their kidney and their heart. So they show up to genetics clinic. We think, well, there's quite possibly an underlying genetic diagnosis. There's something going on in one of your DNA recipes that has caused these symptoms. So we did genome sequencing. It's the biggest genetic test you can do on a patient. You're reading their entire genetic code. And we found a maybe. That's the fancy non-technical term for variant of uncertain significance. It's a maybe, right? We found a DNA change in this patient, and the DNA change was in this gene called ZMYM3. It is one of our 20,000 genes. But we found a change in this gene, and this gene was poorly understood. We didn't really know what this gene was supposed to be doing in the body, which makes it really hard to figure out what it means if you have a change in it. And so we ultimately told this family, we don't know. This might be a diagnosis, but it also might not be. We need to do more digging. So the way that we did more digging is what we didn't do was just sit and wait for another patient to show up in our clinic with the same thing. When you're dealing with genetic disorders, a lot of these are really rare. We're never going to see that second patient walk through our clinic. So we use resources, we collaborate. And so we put this gene into a database called Gene Matcher. It's kind of like a dating service for genetic testing labs and clinics, right? Where we've got a list of the genes that we're like crushing on, that we're interested in. No. And other labs and clinics do the same thing of genes that they're curious about, but they actively want to collaborate on. And when you match, they connect you by email, where we can say, hey, well, our patient has these symptoms. What does your patient have? And they say, well, my patient has developmental delay and this and that. And you go, huh, interesting. We might be onto something. But we can't publish this paper based on 2 patients, because that might just be random chance. And so you wait and you get another match and another match. And in this case, we ended up with 25 other matches, which is a lot. You don't have to have that many to make a diagnosis. But we connected with all kinds of people that had patients that have changes in this same gene, this ZMYM3, that have similar symptoms to our patient. And it was enough to tip us over the edge and be able to say, no, this isn't uncertain anymore. This is the diagnosis for these patients. And we then wrote the paper. First thing we did was to call the patient back, right? And say, hey, we found it. This is no longer uncertain. This is your diagnosis, and here's what that means for you. But the other thing that we did was we published the paper. So then the next time somebody with ZMYM3 changes walks into a clinic somewhere, they don't get that uncertain result. They get their diagnosis right away. And that is one of tons of stories of how HudsonAlpha is helping make new gene disease associations in collaboration with others, because we have so much data on so many patients that have had testing trying to get them a diagnosis. Now, all of these patients that had changes in this gene, they didn't have the exact same change. The way that their ZMYM3 gene was changed was a little different from this patient to that patient, but the impact was the same. The gene was not working like it should. So this is a kind of schematic map of this gene, and you can see all these different points along it. Those are all the places that these patients had genetic changes. They were kind of peppered all around. And the patients didn't all look exactly the same. This is a picture from that paper of the different symptoms and the number of patients that had those symptoms. They didn't all look exactly the same, but there was enough overlap that we were able to say, well, this is not random chance. This is a new disease gene syndrome. much like if you think about any disease out there, it doesn't look the same in every patient. You have variation, maybe due to environment, maybe due to other parts of our DNA that is modifying what a condition looks like. So while we're on this theme of databases, this is another, this is kind of a fun one that, that some scientists at the University of Cambridge put together. And this is an online database called the Unknown. Cute, right? It's a combination of the words unknown and genome. Yeah. And they called it, in one of the papers I was reading, they called it the database of neglected proteins. Oh, I know. But what this database does is it looks at all of these genes and all the proteins they code for, and it gives each one a knownness score. So it measures how well do we understand this gene. And over time, you can see, so the higher the score, so 16 and up, that means we know a ton about it. The scores that are 0 means, well, we know nothing. And you can see over time, we're getting better. We are learning more. There are less genes and proteins out there that we don't understand at all, but there is still work to be done. Just for fun, I decided to put ZMYM3 into Unknown. And this is what popped up. Yeah, pretty cool, right? So we knew very little about ZMYM3 up until somewhere in 2021 or so, when some of this research started to pop up and we started to learn more and more. And these patients started getting connected, which led us ultimately publish that paper in 2023. So that is a story about how we are now able to better diagnose patients with genetic diseases. But the next kind of step after you make a diagnosis is ideally having a way that you can treat genetic diseases. And I want to tell you the story now about some new medications that were FDA approved in the last year to treat a genetic disease called sickle cell. I see some nods. That's a disease that it's still genetic, it's still rare, but is one of the more common rare genetic diseases, if that's possible. But sickle cell disease is a disease of the red blood cells. And individuals who have sickle cell disease, there's a change, a genetic change in their gene, their recipe for hemoglobin that helps red blood cells carry oxygen through the body. And what that means for individuals with sickle cell is that their red blood cells actually have more of this sickled shape to them versus the kind of donut-looking red blood cell that you might have seen before. And what happens when you have these sickled red blood cells is, well, they don't carry oxygen as well, so that causes some problems. They also can get stuck in the blood vessels. They kind of clump up together, and that can cause lack of blood flow and stroke. So this can be a very debilitating condition for people they have pain, they have strokes, and it can be life-threatening for people with sickle cell disease. Now, to explain these new treatments that have come on board that are genetic gene therapy, so using genetics to actually treat the disorder, we need to, for a minute, talk about how our bodies make hemoglobin. So hemoglobin, we actually have a couple different forms of hemoglobin that our bodies make. And there are more than 2, but for simplicity, we're only going to talk about these 2. Our bodies have a recipe for making something called fetal hemoglobin, and also a separate recipe for making adult hemoglobin. And as you might imagine, our bodies typically make fetal hemoglobin when you're a fetus, right? And then sometime right around birth, your body will stop making the fetal hemoglobin and will start on-ramping and making adult hemoglobin. And sickle cell disease is caused by genetic changes in the recipe for the adult hemoglobin. And that's going to be important in just a second. So I told you there are two new treatments that have very different processes, but the impact on the patient is actually really similar. The first of these new medicines is this medicine right here, Casgevy, and it functions, this is a gene therapy that functions like molecular scissors. It's a CRISPR-based therapy, and what this medication does to the patient's red blood cells is it comes in and it actually cuts out, turns off the switch that would be telling the body to stop making fetal hemoglobin. So now you have the situation where the body doesn't have that off switch anymore. And what that means is that then that individual can restart making fetal hemoglobin even after birth. And fetal hemoglobin is really good at carrying oxygen. And so now they're still going to be making those sickled red blood cells. It doesn't make them not do that, but they're making enough fetal hemoglobin that it dilutes it and can ameliorate those symptoms. So that's drug number one. The other medication that was approved, and both of these went through clinical trials, they were proven to be safe and proven to be effective and are now able to be prescribed for patients with sickle cell. The other disease, or the other medication, is this one right here, Lyfgenia. And Lyfgenia is a gene therapy as well, but it actually is carrying in DNA inside a viral envelope. So hijacking a virus system, take out the bad stuff, put in the gene recipe that you want. In this case, it's the gene recipe for that adult hemoglobin, but the version that works. And you are introducing that into the system. And again, you're not taking away this non-working copy, but you're introducing a copy that works, and you're able to make functional adult hemoglobin. Again, diluting the sickled versions and cutting down on symptoms. So impact is the same, but two very different mechanisms to get to the same outcome. Both of these are medications, though, that are not insignificant. And so when we think about, oh, that's amazing, I bet this is getting rolled out to every patient, of which there's like 100,000 in the US with sickle cell, it's not. Right now. These were just approved. And if you put yourself in the shoes of the patient, there's a lot to actually weigh here. Now, on the positive side of things, both of these treatments are believed to be kind of one-time cures. That what the cells that are getting these gene therapies are the blood cell stem cells. They take those out of the patient and then fix it and put them back in. So it actually cuts back on having to find a match like you would if you were doing a bone marrow transplant. In this case, the patient is being their own match, which is good. And by fixing it in the stem cells, you would assume that going forward, all of those red blood cells are going to have this change and be not causing the sickled red blood cells. However, stem cell transplants are not a walk in the park for these patients. It's a year-long process where they have to actually undergo chemotherapy, which has its own set of side effects. And these drugs are expensive, you can imagine. And at this point, as with anything new, insurance is, your mileage will vary, right? And so for patients that are looking at this, they have to weigh, what does my disease look like? What are the symptoms? What are the risks of living with sickle cell disease? And what are the risks involved in undergoing these treatments? And that answer is going to be different for different patients. But these are fascinating avenues, and it's exciting to have options that are not just treating symptoms, that are actually getting at the root cause, and also thinking about how you can learn from this and think about how that might get applied to other types of diseases out there as well. So if we move away from humans for a moment, I want to tell you some other stories about some genetic tinkering, genetic modifications that have happened that are either approved or working their way through approvals. So we're going to talk about purple tomatoes and short corn. What is this? It's a purple tomato. So there is now, there are seeds. So what I'm about to tell you is actually being marketed to home growers. So if you have a green thumb, if you go and look at your seed catalogs, you might find this that you could get and you could plant at home. These are purple tomatoes that are genetically modified, that have two genes that are added to them that are genes that have come from snapdragons. And these genes increase the production of a deep purple pigment, anthocyanin, that makes them purple. It's kind of interesting. But what it also does is it increases antioxidants to where this tomato has the health benefits as if you were eating a whole bunch of blueberries. It's kind of cool. And Americans eat a lot more tomatoes than blueberries. Also, as a kind of win-win, these purple tomatoes also tend to ripen slower. Is an interesting byproduct of that as well. And so you can imagine in certain situations that also might be a beneficial trait for these tomatoes. So purple tomatoes, they've been approved and are able to be sold, and like I said, they're being marketed currently kind of to that home grower group. The other story that I want to tell you right now is about short corn. And there's been a lot of effort in a lot of kind of agricultural crops that have historically grown really tall to tell them to grow shorter. So things like wheat have been over time, over the last several decades, have been kind of forcibly, they've been forced to grow shorter. The reason for that is there's a whole bunch of benefits of having shorter crops. The big one is wind damage. And that's a big deal. Back in 2020, as if there wasn't enough going on in the world, there was a really bad windstorm that wiped out a whole bunch of the corn belt of the US. And so if you've got shorter crops, they're going to be more resistant to wind damage. You also may have to spray less pesticides and those types of things as well. And so there's a lot of benefits for that. And so there's a number of companies, Bayer and others, that are actively working on thinking about how do we genetically control height in corn. What are those genes, right? That gets back to needing to know the function of genes. If you want to do modifications, you need to know where you're going to go. And what are the switches? And how do we control, we want the corn to grow shorter, but we don't want shorter corn stalks, like the corn itself, right? So how do you get that just in the parts of the plant that you are wanting it to be expressed in? So this is a story that's not across the finish line yet. There's a number of companies working towards this. Again, from a number of different angles, they're attacking this. Potentially adding some switches and genes from other plants, or doing selective breeding, right, and kind of forcing it through a more natural breeding process as well. But this is another area of active research to use genetics to help solve problems that farmers are facing. Next story I want to tell you is about a discovery. Again, this is a HudsonAlpha story about discoveries in the field of pancreatic cancer. And if you know anything about pancreatic cancer, you know that it's not good. I mean, no cancer is good. But pancreatic cancer specifically is challenging to be a patient affected by, to be a loved one of a patient affected by, to be a doctor treating it. Pancreatic cancer is known to be very aggressive. It's known to have to typically be diagnosed late. There's not a lot of early warning signs or symptoms that a person has pancreatic cancer. So usually by the time you are making that diagnosis, it's pretty far along, where your options for treatment are pretty limited. And we've got researchers here at HudsonAlpha that are deeply committed to trying to help solve those problems, thinking about how do we better find out who's at risk for cancer, how do we find out earlier who has cancer that's starting, and how do we better understand how cancers respond to treatment and how to make treatments options better for those patients. So the story I want to tell you is some research that came out of the Sarah Cooper lab here at HudsonAlpha that was looking at pancreatic cancer, and specifically this type of pancreatic cancer. I'll say this once and then I'm just going to say pancreatic cancer from there on out. Pancreatic ductal adenocarcinoma. That's pancreatic cancer that starts in the cells in our pancreas that are responsible for making enzymes, and the enzymes that we need to break down the food that we eat. So those are the cells that are turning into cancer in individuals with PDAC. And this is the most common type of pancreatic cancer. So if you hear someone talk about someone having pancreatic cancer, this is probably the type that it is. Pancreatic cancer, like I said, it's aggressive and it's often diagnosed late. The 5-year survival for someone diagnosed with pancreatic cancer is 11%. Yeah. Super important to be researching. So here's what was learned this year in the labs of HudsonAlpha. Our research teams discovered that there's a gene, it's a long string of letters and numbers right here, ANGPTL4. It's one of our genes, that for cancer cells that have this gene turned up, if you think about a switch that would be like a volume control of how much of this gene, how much of the protein it makes is being made, this has the volume all the way up, right? So cells that are making a lot of this protein, that this gene is being used a lot, have worse survival. So when this gene is turned up, patients do worse. So they saw that by looking at a whole bunch of patients and looking at patients that had poor survival, patients that had less poor survival, and looking at what's different in how their DNA is being used. But the question is why? So here's a picture from the paper that they published, and this is a survivability curve, and this is showing that patients that have the volume turned up on this gene, those are the red bar, versus patients that have the volume turned down is the blue bar, and you can see the survivability is better in those that have this gene turned down. So that's an interesting finding in and of itself, but then it immediately makes you want to go, well, why? What is it about this gene that is controlling something that makes patients do better or worse with this cancer diagnosis? And what they found by doing a set of follow-up studies is that, let me go ahead and get this all the way up there. They found that overexpression, so turning up this gene, allows cancer cells to escape the effects of chemotherapy. So chemotherapy does not work as well. That the typical signals that would make those cells die, it ignores them. And the cells survive longer when you really don't want them to. Those are not cells you want to stick around. And so this kind of complicated chart right here is showing what happens when you give cells different amounts of this chemotherapy right here. This is the most common type of chemo that's used. And the part I want to point you to are the kind of teal triangles. So these are cells that have that gene, that ANGPTL4, turned up. And you can see that the viability, the survival of the cells, are increased in those compared to the other types of cells. So turning up this gene makes chemotherapy not work as well. They also figured out that, and we already knew that genes don't work alone, that our genes interact with each other in really interesting ways, and that there's actually some other genes that seem to play a role as well, that either the ANGPTL4 is kind of impacting them or vice versa, but there's something interesting happening between the set of genes. And they discovered that for these other genes that I'm going to put up on the screen in just a second, if you turn down those genes, if you knock down those genes, you can actually reverse the effects of ANGPTL4. Yeah. You can see where we're going with this. Like that points to potential ways that by understanding the mechanism that we can use that to improve outcomes, that you might could think about therapeutics or additional medications that would help knock down some of these other genes that will make patients potentially respond better to chemotherapy. And so what they saw was they had a set of cells, and this is a chart showing how much ANGPTL4 is being made. And so you can see in this set of cells, compared to kind of your normal control, these are cells that have that gene turned up, and they're making a whole lot of that protein. In this situation, we have a set of cells that have that ANGPTL turned up, but they have this other gene called APOL1 turned down. And look what happened. The ANGPTL4 dropped, even though technically it's turned up. This other gene was reversing that behavior. So that was one of the genes, and there was another gene called ITGB4 that had a very similar effect. So better understanding of what's happening inside these cancer cells in the landscape of how they respond to chemotherapy, and thinking about how we can use that information to our advantage. So that was a really heavy story, right? Pancreatic cancer is certainly not a happy topic. This one's a little bit more fun. Based on findings from this past year, we now know that vampire viruses exist. Scared? Yeah. Yeah, go on. So, in case we all need a refresher on what vampires are, these creatures that bite the neck. So we're not talking about Dracula here, but we are talking about something biting the neck of something else. And I would like to introduce you to this little guy right here named the mini flare vampire virus. And not only was the mini flare discovered this year, the concept of vampire viruses is a brand new concept. And part of what I think that makes this story extra cool is that this discovery actually happened by undergraduate students kind of on accident. There was a group of undergrads up at the University of Maryland. So these are not PhD scientists. These are your regular college students that are doing an undergraduate research project. And their project, what they were trying to do was to identify new types of bacteriophages. Big word. Bacteriophages are viruses that infect bacteria. So they were out there taking soil samples. There are tons and tons of types of bacteriophages, like one of the most populous things on our planet. And so there's a whole lot of them that haven't been identified and cataloged, so that's what this research study was all about. Bacteriophages are viruses that infect bacteria. They kind of look like this, where they have a head up top that's got DNA or RNA inside of it. They've got this neck, and then they have these appendages and pins that allow them to stick to the bacteria cell and inject their DNA down into it. The life cycle goes something like this, where they inject their DNA, the DNA gets transcribed, translated, gets they create a whole bunch of new baby bacteriophages that eventually erupt out from the bacteria cell. Yeah, this is Dracula kind of stuff. So these students were trying to identify new types of bacteriophages, and they did. They identified this guy, this big bacteriophage right here. They identified it, and they very creatively decided to call it the Mind Flayer. I guess they've been watching a whole lot of Stranger Things, right? So they named it Mind Flayer, but they got some interesting results and thought that maybe their sample was contaminated because their data had some DNA from something else in there. They thought it was a pure sample. So they put the sample under an electron microscope, and they find this. They find the phage that they thought they were identifying, that they've now called the Mind Flayer here, with this other tiny bacteriophage clamped onto its neck, hitching a ride, in and out of these bacterial cells. And so they then affectionately named him the Mini Flayer, right? So they got a 2-for-1 special in this discovery. And but what's fascinating, something like 80% of the bacteriophages of these Mind Flayers in the soil that they looked at had one of these attached. So this is a major thing for these bacteriophages. But also the kind of creepy part is they found some that didn't have this vampire virus attached, but it still had the bite marks of where one, at one point it had one attached, and I guess it got rid of it, shook it off. Vampire viruses. So who knows? We'll see how many others of these pop up now that we know that this is a thing that happens in nature. And then finally, here is my final story to leave you with tonight. The story of human migration as told by head lice. For starters, you are welcome that I did not create a slide that this is a cartoon. You're welcome. When I went Googling and went and found my images for tonight, I saw a lot of things that I could have subjected you to, but I didn't. But I'm guessing you know what head lice are. Some of you might be itching your scalp as we speak, just thinking about it. These are blood-sucking parasites that live on human scalps that really can't live otherwise. And so over the course of human history, the migration and evolution of lice have come alongside humans. And so we can learn some things from these lice about our own history. And so we have. So scientists have gone all across the globe and taken samples of lice. 274 of them, to be exact, from a whole bunch of locations all across the globe. Current lice as well as archaeologic lice, that's a thing, and have done a whole bunch of genetic research looking at how the DNA of these lice, right, they're alive too, they've got DNA, how the DNA is different from lice that they've taken from all across the globe. And have made some really interesting inferences, discoveries about what that can tell us about how humans have moved across the globe over time as well. So the take-home points that they found is that there are largely 2 clusters of lice. Things you maybe didn't know that you were going to be learning about tonight. 2 clusters. There's a cluster of lice that are genetically similar that were found in places like Africa and Asia and also Central America that had similar genetic material that are genetically similar to each other. And then there was another cluster that, it still has the DNA of a louse, but was distinctly different from these other ones that you found in Europe and in North America. and that largely speaking, there wasn't a lot of mixing of genetic material between these two clusters of lice. And when you look at that and compare it to what we know about how humans have moved over time, what we can learn is that lice have hitched a ride on human scalps to the New World 2 separate times over the course of human history. That there was one time, a long time ago, that lice moved into the New World from Asia over that Bering Land Bridge and kind of came down, and that's how they ended up in that Central America area. But then also, a completely separate instance of lice moving to the New World happened during European colonization. And that only in North America do you actually see this kind of mixing of these two clusters together, because we've got we have lice from both clusters in North America together, but that has all happened much more recently since European colonization. Wow. Cool, right? So those are my stories. Those are a few of the stories from this year, but I want to kind of think about how fast science is moving now, comparatively speaking to what we were able to do in science decades ago. And really that's being driven by several things. One is better technology. We talked at the beginning about the Human Genome Project taking 13 years the first time, and now we can do it in a day. Massive leaps forward in technology that's letting us have better tools and make discoveries faster. Also the power of collaboration, that all of these studies, all these findings would have taken so much longer, or maybe not even been possible if one person was trying to do that by themselves. And the power of computational tools and databases. It's not just about how science technologies move forward, but our ability to write software and use computers to manage all of that data that's coming out of those machines have made scientific discovery possible, and these things all continue to get better, which is really exciting to think about where we're going to be in another year or 2 years or 15 years down the road. So those are a few of the stories in this year's guidebook. Like I said, this guidebook, it literally hit the digital press a few hours ago. So you can scan the QR code there or up here, and you can go and read some of the other really cool stories that I didn't want to keep you here all night telling you all of them. Save some for later. And as we wrap up, a few other kind of notes. Today is our very first Hudson Alpha U event of 2024, but it's not our last. On your sheet, as well as up here, we've got the dates of other ones coming up in May. I hope you'll come back. We're going to do some more storytelling, that time less about discoveries from last year, and we're going to turn the time clock back a little further and talk about Nobel Prize winners in genetics, who they were, the world in which they worked in, and what they discovered and why it matters. And then later this year, we'll have one in September all about family health history and what it can tell you. So I hope you'll come back for those, in addition to all the other amazing events that Hudson Alpha has to offer this year as well. So with that, I want to say thank you for coming out and hanging out with me on a Tuesday night. And thank you for coming out and supporting what we do in education and connecting with us, and also just supporting everything else we do in education to inspire and train teachers, students, trainees, and the community. So y'all have a great night, and we'll see you back in May.